All right, hello and welcome everyone. Thank you for joining us today for Biocytogen's webinar titled Advancing IBD Treatment, Leveraging Innovative Models and Therapies for Enhanced Drug Development. It's great to meet everyone here to discuss the latest in IBD treatment. Please know that this session will be recorded for those who will not be able to attend live. So I'm excited to introduce our speaker, Dr. Luo Jia, also known as Jack. He's a senior scientist and study director at Biocytogen, where he also leads the disease modeling team. So before joining Biocytogen, Jack earned his PhD in infection, inflammation, and immunity from Rutgers, New Jersey Medical School. He then worked on microbiome therapeutics at Seres Therapeutics, targeting infectious and inflammatory diseases, including IBD. He's also a member of the Society of Mucosal Immunology and the American Physiological Society. So today, Jack will discuss the current IBD treatment landscape, the role of humanized mouse models in research, and the exciting future of drug discovery. So let's give a warm virtual welcome to Jack.
Yeah, thank you for the introduction. First of all, I think thanks everyone for showing up to support our seminar. But today I would like to go over some of the animal models we have and potentially could help with your IBD-related research. Ultimately, I think increase the chance of preclinical drugs to go to clinic. So my talk will be include 4 parts: the understanding of IBD and the landscape of current therapies, also the animal models for IBD research, as well as very brief the future directions. So what are the inflammatory bowel disease, IBD? So in very brief, it includes Crohn's disease, CD, and also ulcerative colitis, UC. So both of these are like mediated by inflammation in the GI tract, and CD can affect any part of the GI tract. Commonly seen at the ileum, the distal part at the end of the small intestine, and also the proximal part of the colon. Usually the deep ulcers occur. You see many affect colon and also the rectum part, mucosal area. It usually have seen the ulcers and also the bleeding. Both UC and the CD could have signs of abdominal pain, diarrhea, and the bloody stool, also the weight loss. Currently, approximately about 0.7% Americans, around 1.6 to 3 million people affected by IBD. Many of the patients actually will require surgery during their lifetime. So that provide a huge unmet demand for the new therapies. I think firstly we would like to talk about some of the pathogenesis aspects of IBD. I think in decades that people gradually discover the multi-hit hypothesis. Because IBD is more like multifactorial disease, involve complex interactions between genetic factors, luminal and environmental factors such as microbiome that trigger appropriate mucosal immune response. Often we saw that loss of epithelial barrier integrity, mucosal inflammation, and also tissue damage in IBD patients. And about a decade ago, NIDDK, they set up an inflammatory bowel disease genetic consortium to advance genetic research in IBD. That earlier time conducted some GWAS study, so have discovered over 150 IBD-associated genes. Here I listed some of them, some representative IBD-associated genes for UC, CD, and also some of them are common in both diseases. Biocytogen has created many of the humanized versions of mice based on many of these genes listed here. I will briefly mention later. It's in the GWAS study. It's about 30 genes specifically for CD and 23 for the UC, and the rest of them are kind of in the common area for UC and CD. As I mentioned, we have created many humanized mice off the shelf for the many of the key genes in IBD-related inflammatory and also regulatory pathways. If you are interested in some of these models, we could help with set up some experiments for testing drug efficacy for your research. Some of the I marked red here. We have set up some of the colitis model, or there are some ongoing efforts for the efficacy data. So we go back a little bit more, is that for the IBD pathogenesis, typically in certain susceptible populations or conditions, the commensal bacteria penetrate the epithelium to either because of the disrupted tight junction of epithelium due to some genetic factors, or there is some other microbial factors to penetrate into the non-appropriate area. So you can see that lower down under the epithelium. At that site, the antigen presenting cells such as dendritic cell macrophage will get activated and produce inflammatory cytokines. Such as L1, L612, TNF, and this will lead to more T cell differentiation and also various of immune responses in the downstream. The other very important aspect is the recruitment of T cells from systemic circulations. This typically people find is mediated by the interaction of integrin alpha beta 7 on T cells with MEDCAM1, which is expressed on endothelial cells. So the T cell can transmigrate through the vessels and into tissue. So that will cause excessive immune response and tissue damage. So many of the current therapies, especially the biologics, trying to inhibit one or multiple these key pathways. So it's very similar to the last slides. Here, just showing some of the current therapy landscape. Include earlier ones, anti-TNF, which is very popular for IBD therapies. So this is more like TNF is blocking TNF to bind on the receptor on other cells, which TNF typically is secreted from T cell and macrophages. And also the JAK kinase inhibitor, which can inhibit JAK-STAT pathway, which is exists in many different important inflammatory signaling. Besides that, also people trying to inhibit the recruitment of either lymphocytes from lymph nodes going to the circulations, or from circulations going to the tissue. So in this way, there's two things very critical. One is the S1P receptor inhibitor, which is inhibit the lymphocytes egress from the lymph nodes to the blood circulation. And also the second one is inhibit the, for example, the integrin alpha beta 7 to egress out from blood vessels to the GI tract. And there are many drugs are in the clinical trials. In the field, people are still keep chasing those existing targets like JAK inhibitors or anti-IL-23. But also there are some emerging targets recent years, like TL1A. I will expand a little bit more later. But it's recent years there are many research and many pharmaceutical companies pursue for these targets in the IBD space. In terms of to translate candidates into a clinical trial, we have to do some preclinical studies in either ex vivo or in vivo studies. In terms of preclinical IBD mouse models, there are generally four categories. Today we're mostly focusing on some chemical-induced colitis model, which is kind of popular and common used, and including the TMBS, also DSS model, also the transfer model, usually CD4 positive T cell transfer model, to more like physiologically mimic the IBD progress. So for the DSS model, typically there is acute versions versus chronic versions. There are some difference, but in general, DSS colitis is more primarily by injury or disruption of the epithelium. So in that way, it's activated a lot of innate response, including macrophage and neutrophils. But in acute colitis model, DSS model, there are some Th1 response there, producing interferon gamma, etc. cytokines. But later on, if you prolong that time for the chronic versions, there are some there's Th2 cytokines, also occurred IL-10, etc. And the TMBS model is more driven by T cell response. I will talk a little bit more about it later. The last one is for the CD4 positive T cell transfer colitis model. So the first one is the DSS induced acute colitis model. This model exists for decades. People trying to mimic some of the symptoms of IBD patients. In here, the acute model we're using is using drinking water DSS, usually 3 to 5%, but in this case we use 3% DSS drinking water for 7 days, and also depends on the test articles. We'll do some of the test article will be required daily dosing or the other regimens. The primary readout for this model is doing body weight measurements, also the disease active index score. The DAI score actually contains body weight loss, stool consistency, and also the blood in stool, and combine all of these three factors together to forming the DAI score. Also, the gross measurements for colon length and weight at the terminal would also read out, and also the pathology assessment for the colon. Optional tests. We didn't do a lot of this in our internal study, but we have these capacities to run a cytokine test, flow test, and also some immunostaining. So in this case study, we use a humanized CXCR2, which is IL-8 receptor, IL-8 receptor beta mice to do these acute versions of DSS colitis model. Basically, we do 7 days, 3% DSS drinking water, followed by a couple of days of regular water. The purpose for this part is we want to prolong a little bit the observation to see whether the mouse recovery is different after we have the treatment. And the readout will be the daily body weight and also the DI score. The terminal analysis would be the colon length and weight and also the histology assessment. As you can see here, you can see here all of the groups except the negative one doesn't have DSS, starting to drop the body weight continuously from around like day 3 to 4. And even you stop the DSS treatment, they still typically the wall still going down for a little bit, maybe 1 or 2 days. And then if we prolong the readout from day 8 to day 12, they actually starting some of the mice or treatment starting to recover back the body weight. The rate of recovery is different between the groups. In this case, group 4 and 5, so the purple and the yellow lines, which has anti-CXCR2, I believe this is antibody treatment, 3 mg per kg concentrations, and 10 mg per kg. We observed some of the efficacies rescue the body weight loss. And also that's also true for the DI score. Here we just show because DI score combine these 3 weight loss, stool softness, hardness, and also the bloody stool score to compose the DI score here. We can see that the group 3, the group 4 and 5 has dropped down the DI score later on in the experiments. We didn't observe significant differences at the gross measurements for the colon, including the length of the weight of colon between the groups. And interestingly is that if you look at the histology, the group 1 is more like a benchmark for the wild-type mice, and the group 3 is CXCR2, 3% DSS. They both show some signs of disruption of the crypt structures, and also there are a large amount of immune cell infiltrations. And the groups 4 and to 6, all of these treatment groups actually show the crypt structure is relatively intact there, and also we didn't observe much of infiltrations into the crypt area. And I switch gear to a little bit on TL1A, which I mentioned in the background. Recent years, there are many companies pursue this target. I think Biocytogen also invests quite a bit on trying to develop some of the models in this context. So why is it important for this molecular? So this TL1A function specifically is a T cell co-stimulator, which is relatively so a lot of times its functional receptor, which is DR3, is occurred in activated T cells. So people trying to work on this area to see whether they can actually block in this process to reduce the inflammation. So Biocytogen has a couple of different versions of TL1A mice. I just list this one as an example. For TL1A, 1A is for the strategy of making these mice is using human exons 1 to 4 to replace the mouse version's exons 1 to 4, which is encoded in the extracellular domain of this gene. And we have an example of this mouse in the DSS model, which is TL1A, IL-23, and IL-12. Why these three are together to humanize these genes? So there are many literatures actually support the TL1A actually had suggested with IL-23 and IL-12 to promote interferon gamma productions. So I think that's the reason behind this mice why they created. And this is more like a modeling data, which is titrate the DSS concentrations, because we did pay attention to DSS concentrations in different humanized mouse models. Pretty much we should run a pre-run for different concentrations of DSS. The susceptibility of different humanized mouse models would be differently. In this case, they use 3, 4, 5% different groups, and we observed that when we use higher percent, 5%, it is relatively closer to in wild type we use 3%. So that which produce about 20% body weight drop. So in this case, probably using a higher percentage would be beneficial to create this model. Also, the DAI score suggests very similar information. For 5%, you get closer to the yellow one, which is the 3% in the B6 wild-type mice. And also, there is a histology assessment for the colon. You can see that G3 to 5, which is much more severe than the 3% of DSS in this triple knocking mice, which is consistent with the previous body weight data. From here, you can also see that it's a dose-dependent trend for infiltration in immune cells and also the crypto loss. So we switched to DSA-induced chronic versions of colitis. So this is more like a little bit more closely to mimic the IBD patients has some injury and recover and then constantly into that cycle. And then this required a lower percentage of DSS. So here we use 2% DSS to experience 4 cycles. Each cycle contains 5 days DSS drinking water and 2 days regular water. The readout is very similar to the acute version. Also has clinical score, body weight, colon measurements, and also the HE staining, as well as I think one of the models, I think we run the serious rat for the fibrosis. This is a wild-type mice data, which you can see that is very similar to the acute model. Is starting to drop pretty early at 3, 4, 5 days. They starting to drop the weight, and in that second to third week is at the peak of the body weight loss. And then they're starting to recover the body weight. So that gives some time windows for the treatment. So the body weight loss and the colon weight typically will increase when you have more like inflammation, and the colon nets will reduce. That's more like of ideal situation. Sometimes you will not observe always like these patterns for these two. And the IgE, the HE staining also showed that the crypt disruption, and also you can see that some mucosal area has a lot of immune cells aggregation there. The serous rat also suggests there are some fibrosis there. We also use TL1A as a mouse model here to create this chronic versions of DSS model. We will have used different percentage, ranging from 2 to 3%. So this model has some of the mortality rate to these mice, and there are some variabilities in this DSS model, which I think we probably want to do some optimizations. And we also observed the colon weight increase overall in different percentage of DSS treatment compared to the control, which is consistent with the wild-type mice data. So the third one is for the TMBS-induced colitis model. As I mentioned earlier, people using TMBS model typically trying to look at some T cell response, especially Th1, Th17 related response. So for this model, typically there is a TMBS sensitization earlier a week ago before you actually put another TMBS into intrarectal injections. But I also saw some people doing without this step, but they could have some difference or make some differences here. The readout is the same, so I'll not repeat again. For the it's also the body weight and colon measurements, those readouts. So again, we're using this TL1A mice, trying to create a model using TMBS. As you can see that the readout, like regardless of the amount of you use, actually, you use 3% TMBS, and then all the volume you put in there, you can create a body loss here. And also there are some of the trends of colon length changes between these groups. And the HE staining also suggests, especially in the 3% and also the 67 microliter and 100 microliters, you pretty much cannot see much of creep structures in 100 microliter model here. And there are quite a bit immune cell infiltrations there. And I think there is an internal study to evaluate anti-TL1A antibody, this PF drug, in this TMBS acute model. Which is give some treatment for -1 to 2 days and 5 days post intrarectal TMBS induction. We saw some efficacy at 10 milligram per kg of this drug, but the caveats for this one is they have quite a bit mortality rate. I think for this one. Either the concentrations of TMBS probably would need to be adjusted accordingly to reduce some of the mortality. So we didn't observe much of gross measurements of colon changes shown here, the weight length and the ratio of those two. The histology of 4 groups, they are reducing some of the immune infiltrations in the G3, so group 3, 10 milligram per kg. It's a little bit harder to tell here because I think the angle of the section is a little different from group 2 and 4. The last one is for T cell transfer model. So recent year, I think people. Sorry, I think there's something here. So recent years, people trying to use this T cell transfer model is because it's more physiological relevant compared to some of the chemical induced models. So the general workflow for this model is using enrich the CD4 positive T cell, especially people will exclude CD25 positive population. So they will use CD4 positive, CD25 negative population to transfer into, depending on the background, they will use rag knockout or the SCID mice. So these mice are immune deficient mice. To create this model will need around 8 weeks to create the colitis model. So this is more like a Crohn's disease model, I believe. The readout is very similar, which is also body weight DAS score, the colon weight and length, HE staining, and some other readouts for cytokine or the fibrosis model. So one of the cases here is using a wild-type mice to isolate CD4 positive, CD25 negative T cells, or just straight CD4 positive T cells. As you can see that if you just use CD4 positive T cells, the red color here, you will not create any weight loss or increase DAS score. Because those T cells actually contain some of the regulatory T cells. And if you use CD4 positive, CD25 negative T cells, they can actually decrease the green line, decrease body weight, and also increase the clinical score. The efficacy study here is one is anti-TNF, the other one is anti-CD4 antibody. So a little surprisingly, the anti-TNF actually does not work well here in this experiments. I talked with internally discussing about this. I think we probably will need doing some other TNF regimen treatment, either adjust dosing or the how many times treatment we had in the existing models. Yeah, I think we also can tell some of the signs from the histologies as well. You can see that in the group 5, there's pretty much no like infiltrations there. But group 4, which is TNF treatment, actually has quite some immune cells occurred in there. I think I reviewed all the models here, the common ones we use here, and I hope to like get you into some of the idea about the models here. The DSS model, the acute model is more polarized to Th1, and the chronic version is more like Th1, Th2. We haven't shown here for the cytokine measurements, but from literature, this has been shown for a long time. The TMBS, if you're interested in T cell immediate response, especially Th1, Th17, we would want to try TMBS model. The T cell transfer model would be more beneficial to mimic the T cell dependent colitis or Crohn's disease model. Right now we're under some development for TMBS induced colitis in the BNDG mice, which is our immune deficient versions of mice reconstituted with human HSC, hematopoietic stem cells, trying to create more like closer to humanized immune system. So these are some of the ongoing efforts for different mouse strains, including those TR1A and IL-23 and IL-12, as well as some of the integrants, alpha-4 beta-7, or the combinations of TR1A and alpha-4 beta-7. Also, it would be very interesting to see some of the data for the TNF mice, because in the presentation I did just for the T-cell transfer model, actually should be very interesting to see whether it has some response in these humanized versions of TNF mice. I think some of the key challenges and also the future directions in the IBD space, I have been thinking a little bit about. I think use some of the big data, because right now is everyone pretty much doing a lot of omics, trying to assess the biology. So using the big data to dissect the this pathophysiology. Because IBD is quite complicated, involving genetic factors, environmental factors, immune factors, and microbial factors. So that dissect that is essential for developing new drugs or therapies. And secondly is the heterogeneity of this disease. I think a lot of genetic differential populations, and also the environmental factors occur in different geographic area, also contribute to the disease. So that's one of the challenges for people trying to develop one drug to try to put in their context to fit all the patient populations there. And the third one is to discover more reliable biomarkers. Some of especially earlier diagnostic biomarkers, and also some of the tissue signatures. One example is for the TNF treatment. A lot of patients actually, over I think 2/3 of the patients probably will lose some of the efficacy or response to this treatment over time. I think probably the strategy will be needs to be evaluated during the course of treatment based on some of the biomarkers when patient respond to certain therapies. And also recent years there are some emerging drug modalities besides those biologics. One of them is the microbiome therapeutics. People has been trying to work on fecal microbiome transplantations for a while, but I think there is still lack of evidence to convince people to see whether this is very effective. And also some oligonucleotide based therapies try to inhibit or block some of the pathways using small fragments of DNA or RNA. As well as some cell therapy ways. I think there are many companies actually now is working on developing cell therapies for some of the autoimmune disease such as IBD. Some of the stem cell therapies, and also some people trying to do the Tregs, trying to increase the tolerance of the patients to certain antigens from some of the autoimmune diseases. Yeah, I think that's all for today's presentations. Thank you for your attention. If you have any questions, please text on the Zoom meeting. Thank you.
So yeah, we're going to jump into the Q&A session. If anyone has any questions, feel free to raise your hand or type your questions in the Q&A window. Thank you. I think we already have a few questions here. I'm able to say.
Yeah, I think I didn't mention much about that model. That is more like a spontaneous IBD model, but it's very closely associated with microbiome. Because if you put L10 knockout mouse into germ-free conditions, it will not have spontaneous colitis. So that one is very useful for if we want to look at some of the microbial factors. I think we did some of the studies in CD34 mouse models. We will have these models, but we didn't do a whole lot of work on IBD currently. But I think there is a plan to humanize using CD34. So I just see reconstituted mice in the IBD context.
So I can add one thing about this model. We may not did IBD on that, but it's the mice is available, and we do have like a complete immunoprofiling from like each time point to show that the expression of the markers are like consistent during, and then we have I think the length of how long the study can go after treatment start or anything need to be done. So we data package, please request through the email, and our marketing team will provide information more detailed about that, the whole reconstruct model slides.
Yeah. I think we probably has done something, but I don't think probably we didn't see much of the efficacy data for this model. But I probably need to double check about it. Yeah, I think that's one of the observations. The inflammation in this context typically shorten the length of the colon. Probably maybe it's because some of the the injury created on those epitheliums. But I have to look at it, exactly the mechanism of the shorten the colon length. Yeah. I believe there are some people doing that, but I don't have much experience right now here. I don't think Biocytogen has been looking at this tyrosine kinase 2 mice in IBD context. Oh, the probably Usha is referred to some of the treatment, I believe, maybe the TNF treatment resistant. So some of the the non-responding or non-remits of the patients, I think is they generate some tolerance for the treatment. Some of them probably involving some immune mechanisms. For example, if you probably maybe generated some resistant populations of T cells when you use some of the treatment, there were some other populations expanded. So making, if you want target one and the other one probably get more, because you see if you look at those charts like Th1 Th17 cells. So sometimes you target one, maybe the other one get more expanded. So probably that's part of the reason, I guess, to some of the resistance to some of therapies. I think it seems like if you use a very high DSS in acute model, they will create some mortality. And also it seems from our internal study here, some of the chronic DSS model in some certain strains, they likely have a little bit more mortality rates than the others. And I think overall, the DSS chronic versions has a little bit more variable compared to the acute one currently in our data.
Any more questions from the audience?
Also, for some who are interested in if you are interested in the model we already have, that would be great. So we already have established. If you have some disease model or like something like modified version of that model, and we may not have at the moment, we're willing to do establish that for you. So you just want to say we're flexible on that, and we can work on together to make it work.
I think that's all.
All right. Thanks, Jack and Leila, Natalie, for answering those questions, and thanks to everyone for engaging questions. I think that's all the time we have for today. We hope you found this webinar informative, and if you have more questions, feel free to contact us through the link I shared in the chat. Have a wonderful rest of the day, and we hope to see you again soon. Thank you.